Hello everyone and welcome to the second episode of this mini series on gene mutations on micro and mol bio with Dr. Sunita Bundale. Hope you had a great festival season. In today's lecture, we will be talking about frameshift mutations. If you like the content of this channel, please like, share and subscribe to this channel. In the first episode of this mini series, we have discussed substitution mutations where one nucleotide is substituted in the place of another, leading to various types of mutations like missense, nonsense and silent mutations. Because the mutations involved only one single base, it is also called as the point mutations. But point mutations need not always be substitution mutations. They can also be caused due to base addition or insertion and deletion of a single base. Let us first discuss base addition or insertion. An addition mutation, also called an insertion mutation, is the addition of one or more nucleotide base pairs into a DNA sequence. If only one base is added, it is called a point mutation. However, the number of base pairs inserted can range from 1 to thousands. For example, if this is a nucleotide sequence, then addition of a nucleotide A to this sequence will change this sequence to the one which you can see on your screen. You can see the letter A highlighted in red, which is the newly added nucleotide. A mutation in a DNA is reflected in the nucleotide sequence of mRNA because it is complementary to the gene. Since each codon in the mRNA is of 3 bases and since the genetic code is without punctuation, the insertion or addition changes the entire reading frame from the point of addition. As a result of that, all the codons change from the point of addition. Let us see what effect this has on the protein sequence. On your screen is a gene with a particular sequence of nucleotides. The transcription results in the formation of an mRNA with a specific sequence of codons. The mRNA is translated to give a protein with a particular sequence of amino acids. In this case, the sequence of amino acids is leucine, threonine, proline, glutamine, glutamic acid, lysine and serine. If there is an addition of a nucleotide A in the 5th position, there would be a simultaneous addition of U in the mRNA. The mutated gene will be like this, with an additional base shown in red. On transcription, the mRNA too will have an additional base U. Since each codon in the mRNA is of 3 bases and since the genetic code is without punctuation, the insertion or addition changes the entire reading frame from the point of insertion. All the codons change from this point of addition. On translation, the protein formed will have a change in the sequence of amino acids in the protein. Since insertion or addition of the base results in the shifting of the reading frame, such a mutation is called a frame shift mutation. It results in the change in codons and therefore a different amino acid sequence of the protein. This protein may or may not be biologically functional. The next type is the base pair deletion. Deletions are mutations in which a single nucleotide or a sequence of DNA is lost or deleted. The number of base pairs deleted can again range from 1 to thousands. For example, if this is the nucleotide sequence, the deletion of the nucleotide C will change the sequence to the one you can see on your screen. Deletions also lead to changes in the entire reading frame from the point of deletion. Let us understand this with an example. On your screen is a gene with a particular sequence of nucleotides. The transcription results in the formation of an mRNA with a specific sequence of codons. The mRNA is translated to give us the protein with a particular sequence of amino acids. The sequence of amino acids is the one which you can see on your screen. If there is a deletion of the seventh nucleotide G, there would be a simultaneous deletion of the nucleotide C from the mRNA. The mutated gene will now start looking like this with one base less. On transcription, the mRNA too will have one base less. Again, since each codon in the mRNA is of 3 bases and since the genetic code is without punctuations, the insertion or addition, in this case, the deletion would change the entire reading frame from the point of deletion. All the codons change from the point of deletion. On translation, the protein formed will have a change in the sequence of amino acids in the protein. The changed sequence can be seen in red on your screen. Since deletion of a base results in the shifting of the reading frame, such a mutation is also called a frame shift mutation. Thus, a frameshift mutation is a genetic mutation caused by either insertions or deletions of one or more nucleotides in a DNA sequence. As a result, the reading frame changes, resulting in the translation of a completely different protein which may or may not be functional. On your screen is a reading frame of triplet codons with an ATG start codon and a TAG stop codon. There is a deletion of one single nucleotide A, as a result of which the reading frame is thrown out of frame and leads to the change in the amino acid sequence from tyrosine and also resulting in a truncated protein due to the stop codon UAA. Let us see another example. Here, the addition of the nucleotide C also results in the shifting of the frame and hence change in the codons and therefore a different sequence of amino acids from the point of insertion, which here is again from tyrosine. Let us now see what happens if there is a simultaneous addition and deletion of two bases from a nucleotide sequence. If from a nucleotide sequence, two nucleotides, in this case AT, are deleted, the changed sequence of nucleotides is thrown out of the reading frame, resulting in a frameshift mutation, which leads to a changed sequence of amino acids in the protein. Likewise, in a nucleotide sequence, if two nucleotides CG are added, the changed sequence of nucleotides is thrown out of reading frame, resulting in a frameshift mutation, which leads to a changed sequence of amino acids in the protein. Let us now see what happens when three nucleotides are deleted. When three bases are deleted, the reading frame does not change and hence no frameshift mutation occurs. The three nucleotides form a codon and as a consequence, the resulting protein has one amino acid less in the protein. Likewise, if three bases are added, the resulting protein will have one amino acid more in the sequence, but there will be no frame shift. The protein may still be biologically functional. What happens if there is a simultaneous insertion and deletion of one base? Such mutations are called Indel mutations. Let us understand this process with an example again. Again on your screen, there is this particular gene sequence. The transcription, as you know, will result in the formation of an mRNA with a specific sequence of codons. The mRNA is then going to be translated to give a protein with this particular sequence of amino acids. Now, if there is an addition of nucleotide A in the fifth position, there would be a simultaneous addition of U in the mRNA. If there is a deletion of the 13th nucleotide C from the nucleotide sequence, then there would be a simultaneous deletion of G from the mRNA. The mutated gene will now look like this with one extra base A, which is shown in red, and one base C, which is not seen because it has been deleted. On transcription, the mRNA2 will have one extra base U shown in red and one base G, which is missing because it has been deleted. Since each codon in the mRNA is again of 3 bases, and we have seen that the genetic code is without punctuations, the insertion or addition or deletion will change the entire reading frame, and as a result of that, all the codons are going to change. On translation, the protein formed will have a change in the sequence of amino acids, which can be seen in red only between the point of insertion and the point of deletion. Since deletion of a base results in shifting back of the frame and thus restoring the reading frame, such a mutation is called an indel mutation. However, before the addition, there is no change, and after the deletion, the frame is restored back, leading to the original sequence of amino acids. The position of addition or deletion of nucleotides leading to frameshift mutations is also very important. If the mutation takes place near the 5' end of the gene, it is more deleterious as the entire protein has a completely different set of amino acids as compared to the wild type protein. On the other hand, if the mutation occurs near the 3' end, only the last few amino acids are going to change, and as a result of that, the chances of that protein still being biologically functional are very, very high. Now, let us try to figure out why and when do frameshift mutations occur. The frameshift mutations can occur spontaneously, that is due to errors during DNA replication, such as accidental insertion or deletion of nucleotides caused by DNA polymerases. This is referred sometimes to as replication slippage. There can also be induced mutations caused by exposure to chemical mutagens like the intercalating agents. So let us now try to understand what these intercalating agents are. Intercalating agents are triple-ringed structures whose dimensions are roughly the same as that of a purine and pyrimidine base pair. In aqueous solutions, these substances form stacked arrays and are also able to stack by insertion between two base pairs. The common intercalating agents are ethidium bromide, acridin orange, acriflavin, and proflavin. Let us now try to figure out how these intercalating agents bring about base insertion. Now, this is a DNA duplex. An intercalating agent like acridin orange inserts itself between two adjacent base pairs of the DNA strand by pushing away adjacent base pairs by a distance equivalent to the thickness of a base pair. Now, if this DNA strand acts as template for new DNA synthesis, an extra base is inserted into the new DNA strand opposite the intercalating agent during replication. And after one more round of replication, during which the intercalating agent is lost, the overall result is a base pair addition mutation. This leads to frameshift mutations, which can alter the reading frame and result in aberrant DNA transcription and replication, as we have seen in the earlier slides. Let us now try to understand how these intercalating agents bring about base deletion. This is a DNA duplex. An intercalating agent like acridin orange inserts itself into the new DNA strand in the place of a base, and then when that DNA helix replicates after the intercalating agent is lost, the result is a base pair deletion mutation. This too leads to a frameshift mutation, as we have seen. With this, we come to an end of today's lecture, which is episode 2 of the mini series on gene mutations. We have discussed frameshift mutations, which are caused due to insertions and deletions of one or more bases. In the next lecture, we will discuss the suppressor mutations, which can be considered to be nature's way of fixing and coping with mutations. Till then, stay safe and have a great week ahead. Thank you.